Good afternoon, everyone, and welcome to AAIC Next, the in-person portion. So what we're going to hear from now is 6 early career researchers talking about their work on various aspects of biomarkers or markers. That's just for Henrik, that bit. Just markers, yeah? So we're going to hear about some fluid biomarker research, some digital marker research, and also some imaging biomarker research. And first of all, we're going to have a non-in-person talk, just because we're having so many technical difficulties, I need to get it out of the way. And this is Tom Brown, who works at the Dementia Research Centre, who sadly can't be with us today, but was so keen to talk that he recorded this for us.
Hello, so my name is Tom. I'm a research assistant on the Insight 46 study. Sorry I couldn't be there in person today, but thank you for letting me pre-record my talk, which is going to be on investigating relationships between white matter hyperintensity volume changes and hippocampal atrophy in the 1946 British birth cohort. So firstly, just as a bit of background, white matter hyperintensities appear as bright or hyperintense lesions on T2 and FLAIR MRI. They are presumed markers of cerebrovascular disease. So they sort of manifest as these bright white lesions that you can see around the ventricles on these scans here. So white matter hyperintensities have been associated with Alzheimer's disease and dementia. So this recent systematic review showed that higher white matter hyperintensity volumes at baseline were associated with an increased risk of cognitive impairment, Alzheimer's disease, and vascular dementia. But it concluded that before white matter hyperintensity volume can be considered sort of reliable biomarker for Alzheimer's disease, more longitudinal studies are required to look at changes in white matter hyperintensities over time in relation to Alzheimer's disease, which is what we're investigating in this study. So a paper from the same cohort showed that greater baseline white matter hyperintensity volume was associated with increased rates of whole brain and hippocampal atrophy, and that the rate of hippocampal atrophy was disproportionate to whole brain. And then in terms of the significance of hippocampal atrophy in Alzheimer's disease, so the hippocampus is a structure located in the medial temporal lobe that's thought to be involved in learning and long-term memory. It's a very plastic structure that is vulnerable to damage from different causes like vascular disease, amyloid-beta and tau accumulation, as well as oxidative and metabolic stress. Hippocampal atrophy has been implicated in the early stages of Alzheimer's disease, so it's thought to be one of the most earliest and significantly affected brain regions in Alzheimer's. And as mentioned in the previous slide, baseline white matter hyperintensity volume has been associated with disproportionate hippocampal atrophy. And then in terms of the actual sort of cohort and the data that we're using, so all participants in this study are derived from the 1946 National Survey of Health and Development. They're all born in the same week and have had regular data collection since they were born. And then in Insight 46, since 2015, some of these participants have taken part in Insight, which is a neurological sub-study of the survey. And then in my sort of data set that I'm using, 309 participants from Insight 46 that had MRI data at 2 time points from phase 1 and phase 2 were included. There's average age of 70.5 and the scans are an average of 2.7 years apart. And we excluded participants with major neurological disorders just because they're likely to have higher rates of white matter hyperintensity volume change and atrophy that would potentially drive any associations in the results. So it's a study of a sort of general population cohort looking at sort of predictors or changes in early pre-symptomatic Alzheimer's disease. And then in terms of the actual imaging variables or imaging analysis that we did, so BMOS was used to calculate the white matter hyperintensity volumes. So it's like an automated method of white matter hyperintensity segmentation. So it sort of draws around or segments the white matter hyperintensities, as you can see on that image, and then have that at 2 time points and just calculate the difference between the difference in volume from baseline to follow-up. And then BSI is used to calculate atrophy. So it basically draws anatomical boundaries around the scan, around brain areas within the scan, and then calculates the shift in these boundaries, and that's how the change in hippocampal volume is calculated. So in terms of the actual research questions, what we're attempting to establish, firstly establish rates of white matter hyperintensity volume changes over the 2 time points in the cohort, and then answer the following questions. So firstly, do baseline white matter hyperintensity volumes predict rate of white matter hyperintensity volume change? Secondly, which is the main question, is there a relationship between rate of white matter hyperintensity volume change and hippocampal atrophy? So sort of hypothesis is that those with a greater rate of white matter hyperintensity volume change will have a greater rate of hippocampal atrophy. If this is the case, is this relationship influenced by levels of amyloid-beta or APOE genetic status or cardiovascular risk factors? And then are there any interactions between white matter hyperintensity volume change and these Alzheimer's or cardiovascular risk factors? And the purpose of that is basically to sort of establish what processes might be responsible for any relationship seen, whether they're sort of Alzheimer's or cardiovascular processes. So in terms of results, firstly, as you can see, the sort of trend in the graph shows the higher the baseline white matter hyperintensity volume, the greater the rate of white matter hyperintensity volume change across the 2 time points. And then in terms of answering the main questions, we found that white matter hyperintensity volume increase is associated with hippocampal atrophy. So a 1 mil per year increase in white matter hyperintensity volume was associated with a 0.014 mil per year decrease in hippocampal volume when adjusting for total intracranial volume, like skull size, basically, age at baseline, and sex. This was statistically significant. So yeah, as you can see in this graph, the trend is generally the greater the rate of white matter hyperintensity volume change, the greater the rate of hippocampal atrophy. This relationship was not significantly influenced by levels of the Alzheimer's or cardiovascular risk factors that I mentioned previously. And there were no interactions between white matter hyperintensity volume changes and Alzheimer's or cardiovascular risk factors in predicting hippocampal atrophy rate. And then in a separate analysis, we showed that 4.6% of variance in hippocampal atrophy rate was uniquely explained by rate of white matter hyperintensity volume change, again adjusting for the same variables. So to summarize, increases in white matter hyperintensity volume coincide with hippocampal atrophy, and this relationship is independent of sex, age, TIV, amyloid-beta, APOE status, and cardiovascular risk. So these findings provide a bit of insight into firstly the longitudinal trajectories of white matter hyperintensities in aging, and suggest that increases in white matter hyperintensity volume may contribute to the development of presymptomatic Alzheimer's disease independently of amyloid. So this sort of adds to the literature that I mentioned at the start, suggesting white matter hyperintensities and changing white matter hyperintensity volumes over time may be a valuable biomarker for Alzheimer's disease in combination with other established biomarkers or clinical investigations. And then in terms of future investigations, I'd just like to firstly establish the sort of longer term trajectory of this relationship, which will become apparent when we analyze the data from phase 3, which is currently being collected, and then to sort of attempt to understand the mechanisms that drive this relationship. So the association between white matter hyperintensity volume changes and hippocampal atrophy wasn't significantly altered when adjusting for amyloid or APOE or cardiovascular risk. So we'd sort of like to investigate further what processes do contribute to that relationship. So thank you for listening. sorry I couldn't be there in person, but if anyone does have any questions, please email me at my email address there, so tom.brown@ucl.ac.uk. Thank you.
So the next talk is Fiona Kinsella. She works in the Huntington's Disease Centre and is giving us her first talk on digital markers. And I'm really quite interested to hear this one because I have no idea what you're doing, so this will be good. Okay.
Hi everyone, my name is Fiona Kinsella. I'm currently a researcher in Professor Ed Wild's Huntington's Disease Research Group, and today I'm going to be introducing you to a breakthrough biomedical research device, the smartphone, which you may or may not have heard of. And with this device, I'm going to be walking you through how it's been instrumental in my work exploring digital biomarkers for Huntington's disease, as well as specifically the digital HD study. So, what are digital biomarkers? Well, they are quantifiable physiological or behavioral data that's measured by digital devices, which can be anything from portables, wearables, implantables, and even digestibles. But today, we're going to focus on two of the most common digital biomarker platforms, the smartphone and the smartwatch. But you may be wondering, there are a lot of different types of biomarkers, and we have some pretty good ones for HD already. We've got some great biofluid ones, some great neuroimaging, so why digital? Well, there's a few things that I think give the classic ones a little bit of a run for their money. One, we have the opportunity for completely remote research, which is convenient for the research participant as well as the research staff. And it's relatively pandemic-proof, but hopefully we don't have to run into that again. Additionally, because most, if not all, of the research is conducted in the participant's own home, we have high levels of ecological validity. We have the opportunity to decrease subjectivity between research raters, as the device itself is actually doing the research with the participant instead of a very biased human. And then, looking further down the line, we have avenues for cheaper drug development by potentially reducing the number of in-clinic visits that are needed, acting as a more sensitive measure and detecting changes earlier to therapeutic interventions, and having the potential to reduce sample sizes due to less variability in the data. But if I'm talking to a room full of researchers, which I think that I am, all of this means nothing without a trial to validate it. So that brings us to digital HD. So digital HD was an 18-month-long observational study that was conducted by UCL and funded by Roche, and I'd love to spend the entire time, hours, going through the miraculous effort that was this study, but we're going to do a little bit more of a taster session today. Spoiler alert, I had hoped to have some results to present to you today, but unfortunately, Roche is hard at work doing a lot of data analysis on the copious amount that we were able to get. So we're going to do a little bit more of an introduction, and then we're going to lead into future directions for the research. So digital, two objectives. One, evaluate the feasibility and validity of smartphone and smartwatch-based monitoring. So what does that mean? Well, does the platform work, and do people actually want to use it? And secondly, we wanted to explore what digital monitoring can tell us about the motor, cognitive, and behavioral symptoms of Huntington's disease. Going a little bit more into the study itself, it was 120 participants strong, 40 healthy controls, 40 pre-manifests and 40 manifests. What did we ask them to do? Well, we had them attend 3 in-clinic visits, one at baseline where they were issued their device kit that had their smartphone and smartwatch, a follow-up visit at 12 months, and then the final visit at 18. But mainly, the bulk of the study was actually in the remote monitoring aspect of it, where we asked for participants to engage with their device kit by conducting active tests at home, which I'll go into a little bit more on the next slide. We also had them participate in passive monitoring, so just putting on the devices and wearing them about. And then the frequency of the tests varied, but were on a daily, weekly, and monthly scale. In terms of the tech specs, we had the 2 core devices, like I mentioned, one being the smartphone with a custom-built Roche app pre-installed on it, and the second one being the smartwatch. And then as a supplementary digital biomarker platform, we also looked at the Microsoft Kinect monitoring for motor disorders, really taking advantage of that motor sensor capability of that device. So here are the assessments. As you can see, there's quite a lot of them, and we don't have time to go through them all. So I do have a device kit with me today. If you want to come find me afterwards, we'll do some digital biomarker research together. But for now, I'll just give a categorical overview. So for the active tests, we had patient-reported outcomes that looked at the behavior as well as mood of participants. We had digitalized versions of Huntington's disease research field favorites, such as the symbol digit modality test and the Stroop word reading, as well as a variety of motor tests. And then here down in the bottom right-hand corner, we have passive monitoring, which again is just putting on the devices and wearing them. And then when the participants came in for their in-clinic visits, they did all of the above, inadvertently did passive monitoring, and then a few more additional tests in front of the Kinect. And for all you tech security conscious researchers out there, the data was originally stored on the devices themselves, but once the participant connected to Wi-Fi, the data was securely transferred in fully encrypted form to Roche's servers and then subsequently deleted off the device itself. I want to stay a little bit more with the Kinect assessment because I think that you'll all be familiar with a smartphone app. The Kinect's maybe a bit more out of left field for you. So this is the user interface, i.e. what you would see when you turn on the computer, open the program, and have a subject within view. And this one, we basically ask participants to engage in 7 brief balance tests, but perhaps more exciting is the output. So this is what we get when we have the output from the Kinect. What you're seeing here is the most difficult task that we asked participants to engage in, which was a balance test where we asked people to stand on one leg for 30 seconds. Very hard for anybody to do. And as you can see, as you increase and progress in disease stages, the ability to engage with the task gets more and more challenging. Of course, these are all just naked eye observations though, so we do actually need to do the data analysis to verify these statistically. So, since we don't have any results to discuss today, I do want to talk to you about the sheer magnitude that was this study, because I think it's important for you to hopefully stay on this digital biomarker journey with me now, as well as in the future. So we collected over 100,000 gigabytes of data, and within that we had 24,432 active tests, as well as 431,456 hours of passive monitoring. So an eye-watering amount of data. And then in terms of what the variables and features we examined, there were over 1,100 of them, which may be a bit of a confusing number when you first look at it, because I know there was a lot of assessments on the previous slide, but definitely not 1,100 of them. And so basically, the magic of digital biomarkers is that from a single assessment, you can have a multitude of variables pulled out of that. So what do I mean? Well, giving an example of one of the motor tests we asked them to do. So this is the speeded tapping test, where participants were asked to tap a virtual button on their phone for a minute, and then we assess their fine motor skills from that. So what kind of features can we get? Well, we can look at a regularity of tapping pattern. We can look at amount of time the finger is lifted in the air, or vice versa, the amount of time the fingers are on the button, which wouldn't take that much data analysis to figure out. But there's really a limitless amount of possibilities. And future directions, I will keep it brief because I know it's a bit bold to talk about those before we have any preliminary data analysis finished, but again, I'll keep it brief. So one thing we're really excited about is the idea of a digital motor score. So what would that be? It'd be a total motor score at home. If you're not familiar with the total motor score, it's a clinical assessment that we use in HD that diagnoses the motor manifest symptoms of the disease. So how would we make it? Well, it'd be a combination of the most promising motor features that we're able to extract from the data. And what would it do? Well, if it worked, it would track motor progression remotely, which could help guide care options, determine research trial eligibility, as well as maybe even be a supplemental virtual visit to Enroll HD, which is the largest observational study that we have in Huntington's disease. So I know that that was a lot to cover in a very short period of time. So if there are just a few things I want you to take away from my talk, it's these. Digital biomarkers, relatively new category of biomarkers, which may revolutionize the way we use big data and research. Remote monitoring is convenient. And if it works, which we hope it does, it would offer reliable, valid, and continuous monitoring of Huntington's disease symptoms. But all I can say for now is stay tuned. Can I just give my quick thank yous to the people and stuff? Yeah, just to shout out if anybody's watching that is going to be mad at me if I don't say thank you. But first of all, just a thank you to the Digital HD participants themselves. The studies ran from 2019 to 2022, so they were asked to keep up high levels of motivation during a very difficult time in modern history. And a thank you to Ed and my colleagues at the HD Center, as well as our collaborators at Roche. Thank you guys all.
So next up, we've got Imogen Swift who works on frontotemporal dementia. She's in John Rora's team, but also works with us in the fluid biomarker laboratory. So I know Imogen well, and she's going to talk about her PhD work.
Good afternoon, everyone. My name is Imogen Swift, and I am a PhD student here at UCL. And today I'm going to be talking about using blood to help treat frontotemporal dementia, a race against time. So in the next kind of 8 to 10 minutes, I'm aiming to convince you of 3 things. Firstly, that we are in an exciting era for frontotemporal dementia research, but we are racing against time. Secondly, that fluid biomarkers are a key avenue to help treat FTD. And thirdly, that these findings could help other forms of dementia and unlocking secrets about those as well. So I thought I'd start with a case study. So this is Rachel. Rachel is in her late 30s. She's a teacher and she has 2 young children. And in 2016, Rachel's dad exhibited strange symptoms and strange behaviors. And this included making obscure financial decisions, but also laughing when his grandchildren were crying. And then later in 2017, he was diagnosed with frontotemporal dementia, FTD. And then with more investigations into that, they found out it was a genetic form of FTD. So what did this mean for Rachel? Well, it meant that she had a 50% chance of carrying this faulty gene. And so she decided to go into genetic testing and went through genetic counseling to find out whether she carried the gene. And unfortunately, she was a carrier of this progranulin mutation, so GRN. And this means she's incredibly likely to develop FTD because it's 100% penetrant. So then what does this mean for Rachel's children? Well, unfortunately, it means for each of them, they have a 50% chance of carrying this faulty gene as well. And sadly, Rachel's family isn't the only case of this, with around 10 to 20% of all dementia cases being FTD and around 30 to 50% of these being genetic. But there is some hope. So in our progranulin mutation carriers, there are a number of pharmaceutical trials that are going on with lots of pharmaceutical companies, shown here, developing interesting therapies. And this is based on this idea that when you have the faulty progranulin gene, you have less of the progranulin protein, so that's PDRN. And to show this in a graph, the pink dots on the left are our progranulin carriers, and the blue ones on the right are our non-carriers, and you can see there's less in the pink dots. So this is a key factor for all these trials, and they're aiming to increase progranulin levels. So then what do we need for these trials? Well, we need effective measures of target engagement, and we need effective measures of treatment efficacy. And this is where fluid biomarkers come in, as well as other markers, of course. So for any of you that don't know what a fluid biomarker is, this is the official definition by Kai Bleno. So this is the objective measures of a biological or pathogenic process that can be used to evaluate disease risk, prognosis to guide, I don't know if I'm missing it there, to guide clinical diagnosis or monitor therapeutic interventions. So that's the general thing. But basically, we're looking for this example in CSF, blood, and seeing if we can find some interesting markers. So I do this as part of the Genetic FTD initiative. So just to explain what GenFET is. So it's an international study. We have sites across Europe and Canada. And we look at 3 different groups of people. So these are individuals who have symptomatic FTD and carry a mutation for FTD. They're also pre-symptomatic groups, so they don't have the symptoms yet, but they do carry the faulty gene. And then we have controls that are family members who don't carry the gene. And these are the 3 key gene groups that we look at. So you can see that progranulin is in the middle there, but we also have C9orf72 repeats and MAPT, so that's tau mutations. And as part of this study, we collect various cognitive and clinical assessments, but we also do MRI scans and collect CSF and blood. So, selecting targets for fluid biomarker development. For my project, I decided to look into the biology behind this. So progranulin is involved in a lot of different pathways, both inside and outside the cell. Fortunately today, I'm not going to talk about all these different things because there's a lot going on. I'm just going to focus on these 2. So these are our granulins and our prosapsin. So firstly, prosapsin. So this is a glycoprotein. It is made up of 4 sapacins. It's a lysosomal protein. And the key thing here is that it's a binding partner of progranulin. So we wanted to measure this prosapsin protein in our biofluids to see if there was any significant difference. And so if we first looked in CSF, this is the kind of window into the brain, and we looked in the GenFi cohort, and I used the mesoscale discovery platform for this. And this pie chart explains the different makeup of our participants. So in the outer pie is the 3 gene groups and the controls, and then the inner pie is the split between pre-symptomatic and symptomatic groups. So the darker color is the symptomatic, and the lighter is the pre-symptomatic. And so the findings from this, it's quite a lot to look at. So I'm just going to highlight these 3 parts. So it's this idea that we have a significantly higher level of CSF prosapsin in our pre-symptomatic and our symptomatic progranulin mutation carriers compared to our controls. So then I also sought to see if we could see the same in plasma. This was initial findings looking in a pilot cohort, so just 24 individuals as part of the GenFi cohort. And this is just the breakdown of the 3 different groups. And here, despite no significant difference between the groups, I think it could be to do with the power. So we're hoping to look in a larger cohort to see if we actually do see this higher level that looks like it might be there. So that was super exciting. So looking back at this diagram, what could this mean? Well, in our progranulin mutation carriers, they have less progranulin, and then they seem to have higher prosapsin. So then the potential uses for this could potentially be a secondary outcome measure for the trials that I mentioned earlier, but also a novel target for therapeutics. And then some next steps. As I said, I aim to look in a larger plasma cohort to see if the power can show any significance, but also looking at this complex and whether this progranulin prosapsin complex has any kind of role in the biology as well. So that's example number 1. Now example number 2 is our granulins, and these are notoriously difficult to measure, but I'm going to give a kind of sneak peek data that we've done very recently. So the granulins, they're the breakdown products of the progranulin protein. So there's 7 granulins, 1 paragranulin, and they have likely roles in inflammation and lysosomal function, but to be honest, we don't know too much about them. So the key question is, do these subunits, you can't quite see it, but I'll read it, do these subunits act in the same way as the full-length progranulin? And so to look at this, we used mass spectrometry-based technique, and I did this with Johan and Sophia in Gothenburg, and this was many months of development and testing, as they are notoriously difficult. And this is just to explain the different kind of nomenclature of the granulins and the paragranulin at the end. And we have some preliminary data where we've shown that we can detect paragranulin, granulin 6 and granulin 7, which was really exciting because we thought we might not be able to measure any. And so we took forward this and we looked in a pilot study. Again, this is only 21 participants, so not super high power, but very interesting to see what we could find. And this again in the GenFi cohort. And so looking at this, I've got some very preliminary findings. So we've got the paragranulin here, and then granulin 6 and granulin 7. And so as you can see across all 3 of these, there are significant differences between our controls and pre-symptomatic progranulin carriers. And we also see a trend between controls and symptomatic carriers in the left 2, and a significant difference in the right-hand one as well. So to me, this suggests that our progranulin carriers do have reduced levels of the granulins as well, but obviously we'd need much more cases to prove this. So looking back at this diagram again, in our progranulin mutation carriers, we have less progranulin, and these results suggest that we also have less of these 3 granulins, on one of them being paragranulin. So potential uses for this, could this be a secondary outcome measure in trials, as if they're trying to increase progranulin? Do we also want to show they're increasing the granulins, which have probably some biological benefit? Also, are these a novel target for therapeutics? And then some next steps. As I mentioned, I'd like to test in a larger CSF cohort to get some power and see if we see similar results. Also to quantify potentially in plasma and cell models to see if there's a difference inside and outside the cell. So then I kind of, towards the end of the talk, I thought I'd think about the bigger picture, as I know not everyone works in FTD. I'm in my own kind of FTD world. So I was thinking more beyond FTD and why this could be relevant to you. Progranulin is associated with a lot of different pathways, and these are linked to a lot of different neurodegenerative diseases, including Alzheimer's, but also Parkinson's and motor neuron disease, etc. but also beyond neurodegeneration. It's been linked with Gaucher's disease, diabetes, kidney disease, but also different types of cancer. So it's clearly a kind of jack of all trades, and I think this research could be really important for a number of different diseases. So in summary, I hope I have managed to convince you that it is an exciting era for FTD research, but for families like Rachel's, we are racing against time, and that fluid biomarkers are a key avenue to help treat FTD, and that finally these findings could help unlock secrets for other forms of dementia and also other diseases. So thank you very much for listening and for these wonderful researchers.
I'm going to introduce Will Coath, who's going to come up and tell us who works at the DRC. Yeah, he's going to come up and talk us through some of his imaging works.
Hi, I'm Will. Yeah, I'm a PhD student at the Dementia Research Centre working on the Insight 46 study, same as Tom Brown, whose talk you heard earlier. And I'm going to be talking about amyloid and tau PET imaging and showing some preliminary results from phase 3 of Insight. So most of you will know amyloid-beta plaque, hyperphosphorylated tau containing tangles and neurodegeneration are pathological hallmarks of Alzheimer's disease, and biomarkers can measure these years, if not decades, before symptom onset in vivo. Positron emission tomography or PET imaging can measure amyloid and tau, and magnetic resonance imaging or MRI can measure atrophy due to neurodegeneration. Sensitive and robust biomarkers like these are needed to understand the disease course early in the disease and reduce sample sizes to detect change in AD prevention trials. So a bit about how PET works briefly. So it starts with production of a radio tracer. That's a molecule which sticks to whatever you want it to stick to in the body, and it's labeled with a radioactive isotope. And then this is injected into our participant here in the PET scanner, and then that crosses the blood-brain barrier in brain studies and binds to your target, so amyloid or tau in this case. And then once it's bound, the positrons will be given off from the isotope and they collide with electrons and create this line of response which is detected and an image can be reconstructed from this. And then we have our PET image. However, this, in order to quantify the image, we need to correct things like the dose and other functions. And so the most common way and simplest way of doing this is to take a section of the time after the dose is injected where there's a pseudo-equilibrium and then just divide activity in the area that you're interested in by a reference region which shouldn't contain the protein target. And on top of this, MRI is collected in almost all PET studies alongside in AD. It's used in the processing of PET scans commonly, and you can get other measures like atrophy rates of the hippocampus, as I mentioned before. Having a combined PET MRI scanner reduces scan times for the participant and also has less radiation exposure to the more common PET CT scanners. So Tom mentioned this before, so I work on the Insight 46 study, which is a sub-study of the 1946 British birth cohort. You can see the distribution of participants in Insight 46 matches quite nicely the distribution in Britain of the population. So there was initially thousands of participants enrolled, and they're all born on the same week in March 1946. And they've been studied throughout their life course. And Insight 46 started up and recruited 502 of these individuals for their first study visit in London in 2015. So, as mentioned, phase 1 of Insight 46 started. Participants around the age of 70, and this consisted of included a for beta-peer PET MRI scan, which is an amyloid tracer. And then the same participants, 369 of them completed follow-up scans with the same radio tracer measuring amyloid. And And then now we are currently recruiting, we aim for 250 of these individuals with a second generation tau PET tracer, MK6240. And recruitment's due to finish in, I think, March 2025 for this. So this just illustrates the time window that Insight 46 is collecting data on these individuals in relation to the prevalence of amyloid positivity in the population and the lower prevalence of a diagnosis of dementia, of AD. So now I'll show some preliminary analysis looking at amyloid neurodegeneration in relation to the tau PET in what we've collected so far. So just a bit of methods on the amyloid and neurodegeneration. So I used a very common cortical composite target region and a common whole cerebellum reference region for this analysis. And then these were converted to centiloids, so it's a standardization where a centiloid of 0 is roughly equivalent to the mean of the control group, whereas 100 centiloid is equivalent to the mean of AD participant group. And then amyloid status can be ascertained. In this instance, we use Gaussian mixture modeling to find a cut point. And people were assigned amyloid positive or negative. And then you can also calculate the rate of change in amyloid between phase 1 and 2 of the study, and also the annual rate of hippocampal atrophy between phase 1 and 2 of the study as well, with the boundary shift integral that Tom described earlier. So for the tau PET, so we enriched the subsample for the tau PET for a higher proportion of amyloid positive and amyloid, people who are accumulating amyloid compared to the general phase 2 population. We have currently processed 80 of the MK6240 tau PET scans and calculated SUVRs in each of the BRAC stages, which are a post-mortem way of describing the spatial, the spreading of tau throughout, starting in the transentorhinal region, and so used anatomical BRAC regions from the MRI for this analysis. So here's some results. So this is the tau PET SUVR stratified by amyloid negative on the left and amyloid positive on the right. So 45% of the 80 individuals were amyloid positive, and as you can see, so the cut points for each of the BRAC regions are on the plots, and you can see in the amyloid negative group there are no tau positive individuals past BRAC 1, whereas in the amyloid positive group there are individuals with raised tau. There are some into the later BRAC stages. There are 3 non-conforming individuals. So these are if they're positive in a tau, if they're tau positive in a later BRAC stage, then they've been tau negative. Sorry, if they're tau negative in a later BRAC stage and they've been positive, then they are classed as non-conforming here because they don't follow the hierarchy. This is mostly due to reduced signal in BRAC2 with this tracer, which is the hippocampus, which is commonly seen in studies using this tracer. So now looking at the different BRAC groups and looking at baseline amyloid in the different groups, so tau negative, BRAC 1, 2, BRAC 3, 4, BRAC 5, and 6, you can see that baseline centiloid. So yeah, elevated baseline amyloid if you have any tau. And the rate of amyloid accumulation was higher in the later BRAC stages, so from 3 and 4 and 5 and 6 onwards. You see there aren't that many individuals in the later Braak stage groups, because this is a preclinical cohort. And hippocampal atrophy rate was also elevated for individuals in the higher Braak stages. So just to summarize, tau PET positivity beyond BRCA1 was restricted to individuals who were amyloid positive 3 years prior to tau scanning, and participants with elevated tau in any BRCA region at the tau scanning at age 76 had increased amyloid already at age 70, and increased rates of hippocampal atrophy, and so neurodegeneration, were occurring at least 3 years prior to tau scanning in individuals with advanced tau pathology. This analysis is the first look at results, sanity check kind of thing, and we'll update and expand in future. Thank you very much to my supervisors, Dave Cash, John Schott, Michael Scholl, and the whole Insight 46 research team, and the participants, of course.
Thank you very much.
Introduce Rhian Convery, who works on FTD and is another example of digital markers coming up, I feel. So thank you, Rhian.
Yes. Hi everyone, my name's Rhian Convery and I'm a PhD student at the Dementia Research Centre. And again, I'm going to talk to you about digital markers, but this time for frontotemporal dementia, which is work I've been conducting as part of my PhD, and is part of a much larger initiative called the EDoF study, or the early detection of frontotemporal dementia. But first, just a very quick background on the staging of FTD, because it is a complex illness characterised by different pathological, clinical and genetic phenotypes. And studies such as the GFAP initiative that Imogen has nicely already described for me has shown that the onset of symptoms can be characterised by any cognitive, behavioural, motor or language change. And as a result of this, the label MCBMI has been adopted to capture this clinical complexity. However, the problem is, at the moment, we don't have really good biomarkers that can detect this onset of symptoms. So there's a need for more sensitive markers in the field. And why might digital remote markers be a good way of doing this? Well, they reduce the need for trained professionals, therefore lowering the costs of assessments. They're potentially more widely accessible and can reach a greater number of audiences, producing greater quality data sets. Testing can be done at home, which in turn increases ecological validity. Digital testing allows continuous measurement, again enhancing our data sets. And some sensors are highly sensitive to small changes, so we can obtain more accurate quantifications of performance, for example, reaction time measurements. And digital testing also allows multi-dimensional data sets where we can track different things simultaneously. So the early detection of FTD, or the EDoF study, is a digital biomarker initiative developed at the DRC, aiming to develop a whole different suite of home monitoring tools that could be useful for measuring clinical and cognitive features of FTD, including cognition, language, activity, and motor function. So the cognitive arm of the study includes lots of different things, which I'll go through quickly. So at the top, we have the longevity app, which is a smartphone application developed by a digital health company called Applied Cognition. And this generates digital biomarkers of different cognitive processes based on passive human-device interaction data. Below that, we also have the Ignite cognitive assessment app, which includes computerized versions of gold standard cognitive tests. And alongside that, we also have a C-Hero Quest, which was initially developed for the early detection of Alzheimer's disease and is a more gamified app available for smartphones that assesses visuospatial memory. And finally, we've also developed a portable eye tracking experiment that can be completed outside of the lab and assesses different elements of executive function and social cognition. We're also working in collaboration with a company called Winterlight that have developed different machine learning techniques to extract important markers from speech samples. We have a Fitbit study as well that monitors different physical activity and could potentially be used as an objective measure of apathy, which is an important clinical feature of FTD. and numerous different assessments measuring motor function through a set of collaborative research studies. So we have a surface EMG machine that measures muscle fasciculations, an upper limb motor function tapping test for the assessment of bradykinesia and coordination, and finally a body-worn accelerometer that fixes to the lower back and can output multiple different measures of gait. Oh, there's also the fluidity study, which isn't digital, but is also a home monitoring study that we're running, and you can monitor NFL, GFAP, and progranulin from a drop of blood placed on a blood card. So we're at various different stages of assessing each of these tools, and some of these assessments have undergone thorough validation, and some are just in the very early stages. So for the remainder of this talk, I'm going to focus on the Ignite computerized cognitive app, as this is something I've been testing as part of my PhD and validating. So, as I mentioned, Ignite is an iPad-based cognitive assessment, and it contains short computerized versions of gold standard neuropsychology tasks. And these primarily test executive function and social cognition, which are key early features that present in the disease course of FTD. And these are just 4 different examples shown here, but there are 12 unique tasks in total, and the whole assessment takes between 20 and 25 minutes. So as part of validating this assessment in healthy controls, I conducted a normative data study. So the aim was to generate normative properties for each of the tests based on age, education level, and sex. And I did this through a remote data collection study. So I advertised the app online and asked people if they had an iPad, please could they download the app and complete the tests from home. And wanted around 200 people from each decade of adult life, aged between 20 and 80. But in the end, the number significantly exceeded this, and we have over 2,000 people that completed the app without any other incentive than helping with dementia research, which was really amazing. Just a little bit of data. So from this study of over 2,000 people, I wanted to look at the effects of age, education, and sex on the data. And these are the 4 different levels of the Stroop task along the bottom. And as you can see, hopefully, when you plot the average reaction times with age, there is an increase in reaction times in the older participants. And you see a sort of negative trend when you look at scores. So as age increases, performance on the task decreases. And this is pretty consistent with what we know about normal aging, and processing speed and executive function naturally declines as we get older. So it's reassuring that the Ignite data is also replicating that. From this, I've developed a normative calculator. So when you input the participant details, so their age, sex, and education, and their Ignite app results, you then the calculator then generates a Z-score, which is derived from the normative sample, which is really useful when looking to evaluate people's performance on an individual level. Additional steps in the validation, looking at test-retest reliability, so what happens to the performance on the app when the assessment is repeated, and also concurrent validity, so how well does performance relate to the gold standard versions of the tasks. And we showed that Ignite displayed great, good to moderate actually, test-retest reliability and excellent concurrent validity. And finally, a sort of important step in any validation process, but particularly pertinent to digital assessments, is analysing the acceptability and feasibility of completing these digital tasks remotely. And the Ignite app was rated favourably amongst healthy controls, and even in older adults as well, which is promising. And some very preliminary data is already showing in our genetic FTD cohort that individuals that carry a mutation but don't have symptoms are scoring worse on some of these tasks than their family members that don't have a mutation. And this is an example of a complex emotion processing task where you have to select the set of eyes that matches with the word in the middle. So just in terms of some other projects that are going on, so we've translated Ignite into 8 other languages, so it can be implemented around the world and we can collect even more data. In the future, we'll look at cross-validating the app with neuroimaging and fluid biomarkers of FTD as well, and also do some comparative studies with other mobile apps that are out there for the assessment of FTD. So some really exciting projects in the pipeline. So that was a very brief overview of just one project contained in a much larger digital biomarker initiative of FTD. But the same, we're hoping to follow the same process with these other tools as well. So rigorously validating each of these assessments, conducting feasibility studies to demonstrate that they are, in fact, it is possible to do these tests from home before testing in larger clinical cohorts. And thank you.
And last but not least, I'm going to introduce Rhiannon Laban, who's going to talk to us about her blood card biomarker work.
So I'm Rhiannon. I am a research technician in Amanda and Henrik's lab, and today I'm going to be talking to you about measuring Alzheimer's disease biomarkers remotely, and specifically optimization of NFL and GFAP measurement in dried blood spots, which Rhian just briefly mentioned. So a bit of background. With the recent advances in disease-modifying treatments for Alzheimer's disease, diagnostic biomarkers have never been more needed, and it's likely that blood biomarkers will fill this need because they are quite easily accessible. Yeah. So why NFL and GFAP as a jumping off point? NFL is a marker of axonal damage, while GFAP is a marker of astrocytic activation. So measuring them in blood gives us an idea of the state of these cells in Alzheimer's disease. They're both really stable proteins and can be measured routinely in CSF and plasma. However, sample collection requires specific training and specialist equipment on site. So the draw of the blood spots is that they don't require this specialist training. Theoretically, patients can collect the samples themselves with a finger prick and drop it onto the blood card, which you see here, allow it to dry and send it by post wherever it needs to go. So ideally, they would be great to use. So the first steps towards using them routinely is optimizing the sample eluting method, or the removal of the sample from the card, measuring NFL and GFAP with Simoa technology, and then validating these results. So what I'm going to show you now is like a jumping off. Work by Yoel and Lara was a jumping off point for me, so which is where I got these conditions, starting conditions for eluting. So I used 2 dried blood spots per condition, 120 microliters of eluting reagent, 2 hours incubation, 400 RPM shaking, and 37 degrees Celsius. So we started with venous EDTA blood, dropped it onto the blood card, allowed it to dry, and then stored it in a -80 freezer until we were ready to use it, where we then thawed it and hole punched some of the samples out. It was a 6mm hole punch because that's what we could get our hands on. We then used 2 spots per eluting reagent, and then these conditions with the incubation conditions. I then removed the elute and put it into a new tube, and then removed the blood spots and put it into a bottomless, smaller tube, which acted as a filter. So when centrifuging them, all of the remaining sample from the spots would be removed and into this final tube. Back to the beginning, from the same whole blood tube, we created some plasma, so we'd have exactly matched plasma to the blood spot samples. These sample types were then plated onto a 96 well plate, along with some calibrators and controls, and run on this lovely machine called a Simoa instrument, which is a super sensitive automated ELISA platform. So the first thing I looked at was the eluting reagent. I used diluent and PBS. The diluent comes from the commercial kit that we would always dilute the plasma in, and PBS is very standard in a lab. So I'll just explain the first graph. So we've got plasma concentration along the x-axis and blood spot or blood card concentration on the y-axis. We do expect the blood card concentration to be lower than the plasma, but if it's a true reflection, then they should correlate. And with the diluent, you can see it's not amazing. It's not really strong correlation for either NFL or GFAP. GFAP's in green, NFL's in orange. But when we look at the PBS, it's a much better result, specifically for GFAP. It's a stronger correlation and statistically significant p-value. So this is what we carried on with then, was eluting in PBS. Then I looked at whether we dilute the blood spot eluent or run it neat. So the standard for the Simoa is to dilute plasma 4 times. So that's where that idea came from. So when we look at the neat, not very good. Again, similar to the first graph, not great. But then we look at the 4 times dilution, and it's really, really good, much better than I was hoping for, to be honest. So a really strong correlation for NFL and GFAP, both statistically significant with a p-value of less than 0.05. So this is really encouraging going forward. Yes. So to conclude, the best incubating conditions that I found to measure NFL and GFAP, to use 2 6mm blood spots, eluting in PBS, and using the 4 times dilution on the Samoa. It has to be diluted, and it's likely that the diluting effect decreases or eliminates any matrix effects that we see. For each assay, it's likely that the type of eluent needs to be optimized. So this is just for NFL and GFAP, but going forward, any other ones, it might not be PBS that's the best. And lastly, incubation conditions need to be investigated. So whether 2 hours at 37 degrees is the best, we'll need to look into. So the next steps that I'm hoping to perform really soon are to repeat the same sample with the same eluting conditions in separate wells to see if it's really repeatable. Looking into the blood spot storage, so we did freeze it at -80, but whether that needs to happen, if it could just be stored at room temperature, that would be even better, eliminating the need for these freezers. And then eventually looking into other analytes that are relevant for Alzheimer's disease and beyond, so p-tau, total tau, and the amyloid-beta ratio. I'd like to thank the people here, especially Amanda for her guidance, and thank you for listening.
Brought to you by dementiaresearcher.nihr.ac.uk in association with Alzheimer's Research UK, Alzheimer's Society, Race Against Dementia and the Alzheimer's Association, bringing you research, news, career tips and support.